Hello everyone, my name is Gülten. I'm joining to the symposium from North Cyprus. I'm a lecturer in Nutrition and Dietetics Department in European University of Lefke. Today I'll talk about consumption of plant food bioactives and understanding inter-individual response towards them, and I'll also talk about approaches towards personalized nutrition in the management of cardiometabolic diseases on this topic. To start with cardiometabolic diseases, these diseases including cardiovascular disease, stroke, and diabetes, type 2 diabetes, and they are the leading cause of death worldwide. There are risk factors associated with these diseases, such as hypertension, obesity, diet, unhealthy diets, physical activity, decreased levels of physical activity. These are all of the risk factors associated with cardiometabolic diseases. Evidence from observational studies are suggesting that these diseases could be prevented through diet and lifestyle modifications. So if we have a healthy and balanced diet, and if we are physically active, from an early age obviously, we do have a chance to prevent and also manage cardiometabolic diseases. It's important to establish preventative strategies to manage cardiometabolic disease prevalence, and also evidence coming from population-based studies and randomized control trials focusing on human subjects suggested that a diet rich in plant-based foods show a beneficial effect on cardiometabolic health. So depending on what we are consuming in our diets, we can change our response to these diseases. Today I'm going to talk about plant food bioactives specifically. These are secondary metabolites found in plants. They are produced in plants against environmental stresses, against pathogens, and when we consume them in our diet, there are studies showing that we can benefit from them. Okay? And depending on their basic chemical structure, these plant bioactives can be divided into different families, and there are lots of types of plant bioactives. Here I want to remind that there aren't a set recommendation or value of dietary intake about the intake of plant bioactives, just like vitamins and minerals, but studies focusing on these plant food bioactives are showing that when we consume them in our diets, we can benefit from them. The most abundant families that we will look at as bioactive compounds are polyphenols. We do have flavonoids and non-flavonoid compounds when we talk about polyphenols. Especially in flavonoids family, flavonols, mostly present in tea, is studied really well in randomized control trials for its benefits on endothelial function. So I can say here that the most promising result about polyphenols are coming from flavonols. Anthocyanidins are usually present in purple or red colored vegetables and fruits, so we can find them in aubergines, we can find them in red fruits, and we also have phenolic acids as non-flavonoids. They are highly present in coffee, in tea, and also in whole grains. We have terpenoids, alkaloids, and sulfur-containing compounds as well in terms of bioactive compounds. And when we look at, let's say, sulfur-containing compounds, we do have glycosinolates that are highly present in green leafy vegetables. And again, there are lots of studies looking at their health benefits. So when we look at the effects of plant bioactives on cardiometabolic health, to start with in vitro and animal studies, We can see that results obtained from these studies are suggesting a variety of biological effects involved in the regulation and maintenance of cardiometabolic health. Obviously, data from in vitro and animal studies are not enough to establish the benefits of these compounds on humans. So we also have randomized controlled trials and meta-analysis of human studies showing improvements in the biomarkers of cardiometabolic risk, focusing on these plant bioactives. However, we still need to study these compounds quite a lot to understand their true contribution to the health benefits and their health benefits associated with their consumption. And sometimes there are studies, that's what we will be focusing on today, there are studies showing differences in their responses. So in a way, we can say that not everyone is equally benefiting from plant bioactives when they are consumed at certain doses. Everyone is responding to these compounds differently. And here, this slide is the key point of my talk. We do see, as I mentioned, we do see inter-individual variability in response to the consumption of food bioactives. And there are different reasons for this, and we need to understand these reasons so that we can understand their role in the cardiometabolic disease management. Once we establish all of these, then we will be able to set personalized recommendations specific to individuals about these plant bioactives. Firstly, we have to focus on their absorption, distribution, metabolism, and excretion. We can see that once they are ingested, not all of these compounds are being absorbed to the bloodstream, and from one individual to another individual, we can see different results. We can see one individual highly benefiting and absorbing a certain plant bioactive to its bloodstream, and then we can see another individual who is not absorbing a certain plant bioactive to his or her bloodstream at all. And the reasons for this can be genetic, it can be gender related, it can be age related, but to focus on genetics and host digestion and metabolism, we can see genetic polymorphisms of enzymes involved in the metabolism of these compounds, and these polymorphisms can create differences in the way individuals are responding to these compounds. We also have gut microbiome, which is really important because we know that everyone's gut microbiota is different, and we know that these compounds, some of these compounds, which are structurally bigger, are not being absorbed to the bloodstream from small intestine, and they do travel down to colon, and with the action of the bacteria present in the colon, they can be further digested. So I can say here that composition of gut microbiome can also affect the way we respond to these compounds and the way we absorb them, and it might be one key factor which creates inter-individual variability between subjects. It's not only metabolism and absorption that we see differences, we also see differences related to cardiometabolic disease outcome, and these disease outcomes are blood pressure, blood lipids, endothelial function, and glucose homeostasis. So there are studies also showing that once these compounds are consumed, we do see improvements, let's say, in blood pressure in certain subjects, or we do see improvements in the blood lipids of certain subjects, but we don't see any improvements in others. So we have to understand their absorption and metabolism and the factors that are affecting these, and also we have to understand cardiovascular and metabolic responses that they are creating. So to briefly talk about personalized nutrition, determinants of inter-individual variability could be used when designing dietary interventions for personalized nutrition, and contribution of the different determinants to health effects produced by food phytochemicals should be fully demonstrated and validated. So all of the factors that I said in the previous slide should be fully understood so that we can target an approach towards personalized nutrition. We have to analyze gene variants related to nutrition and metabolism. We have to look for gut microbiota composition and function. We have to evaluate nutritional status of individuals as well as their disease story. And we have to look for anthropometric data and factors such as gender, age, and ethnicity. So when we put all of these factors in one picture and when we fully understand their contribution to inter-individual variability when plant bioactives are consumed or the response against plant bioactives, then we will be able to use personalized nutrition to say people that you can benefit from plant bioactives or you can't benefit from plant bioactives due to these reasons. The potential health effects could be understood, and the more efficient dietary interventions and dietary habit recommendations could be established when we do have all of these factors studied. In conclusion, the identification of these main determinants that we talked about of individual variation for better understanding of their health effects and development of tailored dietary advice is necessary. At the moment, this talk and this piece of evidence is showing us that we have to move away from one-size-fits-all approach towards science-based, personalized nutrition approach so that we can create more effective dietary advices to manage disease status, to manage cardiometabolic diseases, and focusing on plant foods rich in specific bioactive compounds with a higher impact on prevention of cardiometabolic diseases will be the key thing that we will be doing with personalized nutrition when we understand all of the determinants. Thank you very much for listening to me, and I'm very glad to have the chance to join to the symposium. I hope you enjoyed the talk.